Welcome to Group 2A's Chemical Engineering Senior Lab video. This is Shawn Aselin
and Maria Sanchez,
and we performed the drug delivery experiment. The objective of the drug delivery experiment is to explore what factors affect the release of a drug into the body. We used tartrazine to model our drug, which we infused into hydrogel beads. For our analytical purposes, we are treating the drug delivery process as a diffusion of the tartrazine dye through the hydrogel matrix. To create our hydrogel beads, we use a sodium alginate solution and a calcium chloride solution. We mix a tartrazine dye into the sodium alginate solution to represent our drug. When the sodium alginate is dropped into the calcium chloride solution and exposed to the calcium ions, a hydrogel bead is formed through a process known as cross-linking. Different factors may affect the rate at which a molecule diffuses out of a hydrogel. In this experiment, we tested bead size, mixing rate, and temperature as our variables. We will now briefly go over the procedure for this experiment. The first step of the procedure is to create the solutions. To make the alginate solution, we added 1 gram of alginate to 100 milliliters of a 0.5 grams per liter tartrazine stock solution. We then prepared a 6% by weight calcium chloride solution from distilled water and calcium chloride hexahydrate. The calculations for this solution were slightly complicated by the fact that the calcium chloride already had water molecules in it. In this experiment, we were using a spectrophotometer to measure absorbance. We must make a calibration curve in order to correlate this into concentration of tartrazine in the solution. We mixed tartrazine solutions to several different known concentrations and measured the absorbance. We used Excel to graph this data and get our linear correlation curve.
For bead production, we first begin by pouring our calcium chloride solution into a large weight boat. Next, we use a disposable syringe to extract about 3 milliliters of our alginate solution. We dispense about 60 beads of alginate into the calcium chloride solution, which should take about 1 minute. It is important to avoid dropping the beads on top of one another. Once the last bead is dispensed, we wait about 5 minutes to allow for cross-linking. Different needle attachments will create different size beads. As soon as the 5 minutes have elapsed, we separate the beads from the calcium chloride solution through filtration. When we're ready to begin the experiment, we fill a 150 milliliter beaker with 100 milliliters of DI water. Next, we add a magnetic stir bar and then place the beaker on top of a mixing plate. We set the temperature and stir rate accordingly. We drop the beads into the beaker and immediately take our first sample. The sample is pipetted into a cuvette and measured for absorbance. Afterwards, the sample is returned to the beaker to maintain constant volume. A reading is taken every 5 minutes for the next 60 minutes. After the completion of this experiment, all waste is disposed of in its proper container. Glassware is cleaned and placed on the drying rack. The lab area is cleaned and sanitized.
We tested the effect of 3 different variables on diffusion rate. These variables were bead size, mixing speed, and temperature. We tested small, medium, and large beads at 120 RPM mixing speed and room temperature. In addition to 120 RPM, we also tested the large beads at 240 and 360 RPM mixing speeds. Finally, we tested the large beads at 120 RPM and 40 degrees Celsius. 3 trials were performed for each set of conditions. First, we will look at the effect of bead size. The small beads reach a final tartrazine concentration of about 0.0014 grams per liter. The medium beads reach a final concentration of about 0.0025 grams per liter. The large beads reach a final concentration of approximately 0.004 grams per liter. All the beads have the same concentration of tartrazine, but the larger beads have more mass, so they contain more tartrazine. This is why the larger beads yield a higher final concentration. Next, we will look at the effect of mixing speed. As you can see, the final concentration is not really affected by the mixing speed. However, what is affected is the time it takes for the solution to reach the final concentration. The increased agitation associated with the higher mixing speed causes the diffusion to occur faster. This produces a steeper curve on the graph that approaches the final concentration faster. As you can see, the beads take about 40 minutes to reach their final concentration when they are being stirred at 120 RPM, but only about 25 minutes to reach their final concentration when they are being stirred at 360 RPM. The last variable we decided to test was temperature. Since we are exploring the phenomena of drug delivery, we decided to test the diffusion rate at 40 degrees Celsius, which is close to your natural body temperature of 37 degrees Celsius. We see that the increased temperature leads to a decrease in the time it takes for the solution to reach the final concentration. By increasing the temperature from room temperature to 40 degrees Celsius, we decreased the time it took to reach the final concentration from 40 minutes to 20 minutes. The temperature seems to have significantly affected the diffusion rate. This makes sense because an increase in temperature is related to an increase in kinetic activity of the molecules, which would cause the diffusion to occur more quickly. In conclusion, we find that the final concentration of the tartrazine in the solution is only determined by the bead size. This makes sense because the larger beads contain more dye, so there is more dye in the solution when equilibrium is reached. It is also clear that factors such as mixing speed and temperature may have a significant effect on the release of this drug. This information may be pertinent to a biotechnology company that is looking into developing drug delivery techniques. Experimental conditions must be carefully chosen to make sure that they closely mimic the conditions that would be found naturally in the body. If any turbulence or mixing is occurring in the body, it must also be simulated in any laboratory experiments in order for them to be accurate. Similarly, the results from varying the temperature show us that it is extremely important that any experiments be carried out at 37 degrees Celsius in order to simulate the human body environment. There were a few potential sources of error in this experiment. We noticed that when making the beads, sometimes they would get distorted as we dropped them into the calcium chloride solution. The best way to solve this problem is to hold the syringe as close as you can to the calcium chloride solution so that it doesn't drop very far. Cross-linking time is also an important factor. We waited 5 minutes after dropping the beads into the calcium chloride solution to allow them to cross-link. More time would result in more cross-linking and a stronger hydrogel matrix. In one instance, we used a different type of filter. The calcium chloride solution took much longer to filter through, and the cross-linking time was essentially increased by about 2 minutes. Significant variations in the cross-linking time may lead to experimental error. Additionally, when we dropped the completed beads into our beaker of water, there was sometimes some tartrazine solution on the outside of the beads. This would immediately be mixed into the solution and would show up in our initial reading. In order to prevent our initial readings from being so erratic, we could add a step in the procedure where we rinse the beads before dropping them in. This should get rid of any excess tartrazine on the outside of the beads and hopefully yield initial readings that are close to zero.
Thank you and good luck, future seniors.